everyone. Today we will be studying about the development of endosperm of the first chapter, that is the development of an endosperm. The first question that arises in the mind is what is meant by endosperm. So I think that you all remember that pen which we have discussed in the double fertilization, that is nothing but the primary endosperm nucleus. So that is only the triploid primary endosperm nucleus respectively divides, that is repetitively it divides mitotically to form the nutritive tissue, that is nothing but the endosperm. Clear everyone? So this, what is the definition of endosperm? They are trying to say as that nothing, that is the triploid primary endosperm pen, that repetitively undergoes the mitotic division and that forms a very nutritive tissue, that is nothing but the endosperm. So endosperm is the nutritive tissue for the nourishment of the developing embryo. So for the providing the nourishment to the developing embryo, the primary endosperm does the work over there. So next is what we are going to study about, that is the primary, that is this endosperms are divided into three types on the basis or on the mode of development. So there are three types as you can see, that is nuclear type, cellular type and halobial type. So first type is the nuclear type. So in this nuclear type what happens, that is the primary endosperm nucleus, that is the pen you can say in a short form, that repeatedly divides mitotically without any wall formation, and that to produce the large number of a very important term, that is the free nuclei. That is what happens over here, that is the primary endosperm in the nuclear endosperm, it undergoes mitotic divisions and it results in the formation of free nuclei. One thing you should keep in mind over here, that here the process of karyokinesis takes place. Nuclear division is taking place, means certainly karyokinesis is taking place. Cytokinesis is not being done in this stage. So this what happens, the formation of large number of triploid nuclei that freely suspended takes place over here. So later on what happens, later on wall formation occurs around this nucleus in a centripetal manner and to form a cellular mass. So this is the most common type of endosperm which is seen mostly. So what happens over here, that is the vacuole appears at the center and this vacuole pushes the embryo towards the periphery or towards the peripheral condition. Okay, first this was the oospore, that is which we got at the first fertilization. Next the primary embryo, that is endosperm. What happens over there, endosperm divides and it creates the free nuclei all over, and later on this vacuole which is being generating over, means which is being created over here, pushes the embryo or the oospore towards the at the peripheral condition. Okay, almost you can say mostly this full division that doesn't take place, always it stops at the creation of wall and the empty space is being created inside that mostly. Okay, so next we will see about the cellular endosperm. Okay, that is cellular type we are going to see, that is. So next we are seeing that is the cellular type. In this what happens over there is the division of the primary endosperm nucleus. That is the division of primary endosperm nucleus is immediately followed by the wall formation. So in this what happens is the primary that is endosperm nucleus undergoes nuclear division which are followed by cytokinesis. Means here in this phase karyokinesis was only done, but in this what happens over there, that is karyokinesis and cytokinesis both take place simultaneously. Okay, so therefore the endosperm development occurs in the cellular from rightmost from the beginning only it takes place over, and it is very less observed, and it has been only seen in the dicots. Okay, dicots family. The example I have written over there is balsam. So the structure in cellular type means occurs like this. What happens over here? That is the oospore is formed over here, and later the division occurs in this manner, and next we are going to we see a large that is fully packed of cells. That is the cellular structure. Last one we are going to see about the halobial type. So halobial type, that is, so in this what happens is the first primary endosperm nucleus is the first division occurs is followed by the formation of a transverse wall, and which divides the wall very unequally over there. And what happens over here? So there is two division which is being formed, a smaller one and a larger one. Okay, so both the structure is formed over there. So in that what happens later, that is they have specific name over there. That is the central is divided into larger micropyle. Means the larger one, the large one will be said as the micropyle, and the bottom one means the smaller one will be called as the chalazal chamber. Okay, so in this what happens over there now, that is the development of endosperm occurs as nuclear type. Text is like that of the nuclear division only. It is an intermediate type which is common in Halobia series of monocots. Okay, so it is mainly still in the monocots division only. So you should remember that in the first we have seen about the nuclear type, second we have seen the cellular type, and third is of the halobial type. But all these divisions are of endosperms only, and these three modes depends on the basis of their development only. Okay, so nuclear type, you should remember that it goes repetitive mitotic division, and there is a formation of free nuclei which results in the process of karyokinesis. But when we are talking about cellular type, what happens over there? That is the division of primary endosperm nucleus takes place very rapidly over there, and in this process karyokinesis takes place certainly, but it is being followed by the cytokinesis also. There is a formation of cellular structure cytoplasm inside that. Therefore, it is being said over there now. Now it is being observed in 72 families of dicots. Means you can say that very less family only you can say it is being seen over there. Last one is the halobial type. In that what happens over there? That is the unequal divisions takes place from this transverse section. And what happens over there? Larger one and the smaller one which you get. So larger is called as micropylar and smaller is called as the chalazal. Okay, so in this manner we have completed this whole topic. Next we will be seeing development of embryo. So the process of development of zygote into the embryo is called as embryogenesis. So what is embryogenesis? It is nothing but a process of development of zygote into the embryo. Okay, so this embryo developed in the micropylar end of the embryo sac. So after certain amount of endosperm is formed, that is after the fertilization, the embryonic development starts. So that you can say that the development of embryo starts at that time only, that once the fertilization has taken place. Okay, so after that what will happen is, you can see the diagram, that is a diagram you can see at the left upper corner. Okay, so that is the zygote. Okay, that is the zygote which divides into two-celled, that is the proembryonic state, that you can say as the proembryony or the larger cell. Understood? So in the diagram of B you can see that there are two-celled structure. Okay, two-celled, that is they are called as the proembryony. Okay, and next you can see that the larger one in the B only, see the larger section is called as the micropylar end, and to the upper one that is called as the, you can say as, that is micropylar end, towards the micropylar end, that is called as the suspensor, and that is towards the basal part, that is called as suspensor, and towards the chalaza, means the upper part, is called as the embryonal initial cell. Okay, so to produce this, what will filamentous suspensor, now in the that is the C picture, you can see that a filamentous structure is being produced. So this filamentous structure is of 6 to 10 cell. So this what happens over here, that the first cell of the suspensor, what they do towards the, they push them towards the micropylar end and become swollen, and the function as a hostorian, that H you can see over there, yes, so that H is nothing but the hostorium. Okay, so next the lowermost cell of the suspensor is the hyposis. Next, when you come towards the bottom section in the COL, you can see that is called as hyposis. This suspensor pushes the developing embryo. So the embryo which is being developing inside is being pushed out, is being pushed deeper and deeper in the endosperm for the proper nutrition. Endosperm and embryo, the relation between them is that to get a proper nutrition, only the endosperm endosperm has to perform that function. Okay, so now once the suspensor has developed, what will happen? You can see that filamentous structure. Now that filamentous structure in the picture D, see that EHS आपको ऐसा कुछ दिख रहा है वहां पे, that is nothing but the embryo. Now this embryo will enter inside that endosperm, and what will happen? That will apical cell of the two-celled pro-embryo undergoes a transverse and vertical division at right angles to each other to form an octate stage. ऑक्टेट स्टेज में क्या होगा वहां पे, अभी एक आप देख सक रहे हो, नेक्स्ट आप देख रहे हैं यहाँ पे एफ डिविजन, वी कैन से ई, वी विल सी ई इन दैट व्हाट हैपेंस, यू कैन सी दैट इज a heart shaped structure has been formed out there. So this 8 cell of the octate proembryony undergoes many divisions and it forms a spherical mass of cells. So later on in the eole, see at the top, that is called as the heart shape. Okay, and then it turns into the horseshoe shape. So therefore after that what happens over there, they form a embryonal axis, that is plumule, radicle and 2 cotyledons. In the diagram G you can see 2 cotyledons at the top position, it is being shown. Next is plumule, you can see. Next is what happens over there, and a radicle and a single cotyledon is being seen. 2 cotyledon will be seen in dicots and 1 cotyledon will be seen in the monocots. Okay, thank you.